Welcome to the Week of Evolution webinar series. We, as Boğaziçi University Science Club Trio of Evolution Committee, collaborated with many scientists who are experts in their fields to present an evolution theme series to you. We wanted to use the opportunities of the online platform to meet with professors all around the world.
Our mission was always to spread accurate scientific information and create platforms where science can be freely discussed and told. When the conditions are present in our country are taken into account, it is even more crucial to do so.
Therefore, we would like to restate the necessity for free academy as we still protest and stand against the appointed rectors and their undue sanctions.
Both university members have been standing against this anti-democratic situation for more than a year.
Aside from that, this year we will have 9 seminars between 21st of January and 7th of February. You can also follow our social media accounts to be posted about calendar.
Today we will welcome Professor Joshua Payne from ETH Zurich. After he is done with his talk, we will have a short Q&A session. You can ask the questions through chat, and our team will edit and direct them to Professor Payne. Welcome again, Professor Payne.
Yeah, thanks. Happy to be here. Can everyone hear me all right?
Yeah, we can hear you now.
Great. Hey, I wish I was visiting you all in person. I've been to Istanbul once about 10 years ago, and I had a wonderful time there. My best memory is actually just like a small one. I absolutely loved kumpir. That stuff is like, as an American, I love baked potatoes, and you guys do that just perfectly. I also went to a Beşiktaş game, and that was a lot of fun. So I wish I was visiting you in person, but this will have to do. And thank you very much for inviting me today. So the title of my talk is Mutation Bias and Adaptive Evolution. And I'll start out just giving you an outline of what we're going to talk about today, right? So I think that it's important to give you some background on this problem. So why are we concerned with mutation bias and how it influences adaptive evolution? And then I'm going to talk about 3 papers that I've been part of that investigate the role of mutation bias in adaptive evolution. And excitingly, one of these papers is actually going to come out next week in PNAS, so it's quite fresh. And that just is a nice opportunity to talk about some really new research. And then finally, discussion with you, right? Good. So let's start with some background. So I think that we all appreciate that mutation is a stochastic process, right? But what is perhaps less appreciated is that mutation is a biased stochastic process. So for instance, mutation rates vary across the genome, depending on, say, the origin of replication or on the transcription level of genes. And mutation rates also vary amongst different classes of mutation types. So for example, one of the most straightforward distinctions you can make with regard to mutation types is that of transition mutations and transversion mutations. So transition mutations are between a purine and a purine, or between a pyrimidine and a pyrimidine, whereas transversion mutations are between a purine and a pyrimidine, or vice versa. Now, if you just look at this little diagram here, one thing that you'll notice is that it doesn't matter which nucleotide you're looking at. Obviously, there's always 3 mutations away from that nucleotide, and it's always the case that one of those 3 is a transition, whereas the other 2 are transversions, right? So if mutation was an unbiased stochastic process, a null expectation would be that you would always observe 1 transition for every 2 transversions among just neutrally occurring or spontaneously occurring mutations. But that's typically not what you see. So here, if you look at data from 5 bacterial species, where on the y-axis you're looking at basically the frequency with which a mutation spontaneously arises, grouped according to the 6 possible nucleotide mutation types, that's on the x-axis, and then further grouped according to whether those mutations are transitions or transversions, you see that transition mutations are the most frequent. right? So they're occurring much more frequently than you'd expect, given this sort of 1 out of 2 null expectation. And this isn't something that's specific to bacteria. So we find such transition bias across the tree of life. So here are just a few figures that are illustrating this. These are from different studies, so they're not immediately visually directly comparable, but please trust me when I say that all of these figures are uncovering or reporting transition bias in species as different as yeast, plants, animals, and viruses. All right, so transition bias clearly is part of the biased stochastic process that is mutation. And the question is whether or not this bias in the generation of genetic variation can actually influence which genetic changes drive adaptation. So this turns out to be a very controversial question. And this controversy actually has its roots all the way at the base of evolutionary theory. It has its roots in Darwin's Origin of the Species. So in the original editions of the Origin, I should say, Darwin put forth sort of 2 scenarios by which evolution can proceed. And each of these scenarios involved wolves. So in scenario 1, he envisions a large group of wolves that vary in phenotype. So some are bigger, some are smaller, some are stronger, some are weaker. And all of a sudden, there's some environmental change. And that environmental change makes their prey more scarce. And because of this environmental change, it's only those stronger and faster wolves that are actually able to catch their prey. And so the phenotype of the wolf population, pushes towards this faster and stronger phenotype over time for obvious reasons. Now, what is important in this formulation of evolution is that evolution proceeds via selection on standing genetic variation, right? Those variants were already present. That's scenario 1. Now, scenario 2, wolves again, but in this case, all the wolves are the same. Same exact wolf, loads and loads of them. Then all of a sudden, one wolf has a mutation, and that wolf, say, is faster or is stronger, whatever, and it's better able to capture its prey. And so again, the population is going to start to move towards this more adaptive phenotype, right? This phenotype that's better able at catching prey. But what is different in this scenario is that evolution did not proceed via selection on standing variation, but rather via selection of a new mutation. So what differentiates these 2 scenarios is the importance of standing variation versus new mutations, right? And the reason that this controversy is already at the root of evolutionary theory, and the reason that I put the 6th edition of the Origin here, is that it was in this edition where Darwin disavowed this 2nd scenario, right? He said, in former editions of this work, I sometimes spoke as if this latter alternative frequently occurred. So the latter alternative being this 2nd scenario in which new mutations matter, right? And then this view that selection is sort of the creative force in evolution, that selection is always working on standing variation, is the view that was adopted by the architects of the modern synthesis. So for instance, you have Ernst Mayr saying the synthesis was a reaffirmation of the Darwinian formulation that all adaptive evolutionary change is due to the directional force of natural selection on abundantly available variation, right? It's all about this gene pool where you have like all the kinds of variants that you could possibly need already present. And this idea was further championed by other sort of towering figures of the modern synthesis, such as Haldane and Fisher, who conceptualized evolution as a process of shifting gene frequencies. So you have this abundant standing variation, and it's selection that's really the driving force in changing gene frequencies. And mutation is like this weak, ineffectual background operator, because mutation rates are so small relative to selection coefficients. Like, how could any bias in the mutation process possibly influence like adaptive trajectories? So that just kind of seemed impossible in this conceptualization of how evolution worked. But later on, you have these other massive names in evolutionary biology, like Kimura and Gillespie, working with models that weren't so concerned with shifting gene frequencies. These models are so-called origin fixation models, where you have a population that's monomorphic, so there's no standing variation, and then a new mutation arises, and it either goes to fixation or it goes to extinction before another mutation comes along. Right, so this is sort of implicitly going back to Darwin's second scenario, right, where it's this new mutation that causes this new phenotype in this wolf that is either advantageous or not. And in this origin fixation regime, I don't think it's very difficult to imagine that if there's a strong bias towards a particular mutation type, then that bias could bleed through into which genetic changes actually drive adaptation, right? Because this is like a first come, first served process. If something comes along just because it's more frequent, it doesn't have to compete with something that might be higher fitness but less frequent, right? It's just good enough. And so it can go to fixation. So this is this sort of the background of this controversy. And I'd like to highlight that this controversy continues to this day. So if you want to read some modern evolutionary biologists that really do not like the idea that mutation bias can influence adaptive evolution, I recommend this piece by Svensson and Berger. And if you read that, I highly recommend the, I think, 12-piece blog post that Arlen Stoltzfus wrote in response to this that absolutely eviscerates these arguments in a characteristically incendiary way. Additionally, if you're interested in sort of understanding more about this history, going all the way back to Darwin, I highly recommend this paper that actually just came out by the philosopher John Beattie. It's a wonderful exposition of just the roots of this problem. Now, I personally have never been motivated by controversy. Like, I don't really care if some old figures in evolutionary theory thought one way or the other. I care what models tell us and what data tell us. That's what I care about. I have no skin in the game when it comes to this controversy. I'm just interested in actually sitting down with models and sitting down with data and figuring out what they tell us about when and where and why mutation bias can influence adaptive evolution. So the final bit of background here is the first piece of theory that suggests that mutation bias can influence adaptive evolution. So like so many theories in evolutionary biology, this one is based on a two-locus, two-allele model. And in this two-locus, two-allele model, you have two adaptive peaks, one at genotype little a big B and another at genotype big A little b. Genotype little a big B is selectively disfavored, so it's the lower peak, but it's mutationally favored. There's a higher mutation rate to that peak. Whereas big A little b is selectively favored. It has a higher selection coefficient. It's the higher peak in the landscape, but it's mutationally disfavored. So the mutation rate to that peak is lower than it is to the other peak. So the question is, given this setup, like, are there ratios of these two mutation rates that bias evolutionary trajectories toward the selectively disfavored peak? And indeed there are. So on the x-axis here, you're looking at exactly this ratio, which you can just call mutation bias. So this is the ratio of the rate to the selectively disfavored peak relative to the rate to the selectively favored peak. And on the y-axis, you have the bias and outcome. So how often do you end up on the selectively disfavored peak? That's the going up above 1. And how often do you end up on the selectively favored peak? That's below 1. And you're looking at this relationship for different population sizes. So as you read these lines from top to bottom, the population size is increasing. So what's quite clear from this plot is that indeed there are ratios of these mutation rates such that the expectation is that evolution will converge on the selectively disfavored but mutationally favored peak. The other thing to take away from this plot is that one of the most basic population genetic conditions you can think of, namely population size, has an influence on this process, right? So the larger the population size, the less likely mutation bias is to steer the evolving population toward the less adaptive peak. And the logic here, so if we're thinking about this first come, first served process, this origin fixation dynamics, right? Population size influences mutation supply. So the more individuals that you have, the more mutations you have in that population. And when that is true, then even if you have a strong bias towards a selectively disfavored genotype, that bias will be outweighed by the sheer number of mutations that are happening, which pretty much guarantees the arrival of the selectively favored mutation, right? And that selectively favored mutation will be able to outcompete those mutationally favored mutations. Okay, so this is sort of the first theoretical evidence that mutation bias can influence adaptive evolution. This is a wonderful paper. And one of the things that's cool about this field, if you're interested in getting into it, is it's kind of like a one-stop shop. Arlen Stoltzfus has been standing on this hill of mutation bias for more than 20 years now. And for the longest time, nobody was listening to him. He was like a heretic in evolutionary theory. And it's only now that his ideas are starting to get traction because we can actually test them with experimental data, as I'll show today. So if you're interested in this, read Arlen's work because he's done more than really anyone in the field on this topic. Now, this sets the foundation, right? And my PhD student, Alejandro Cano, was interested in this mutation bias literature. And he had read this paper and he thought, okay, well, the study of adaptive landscapes has come a long way since, the early 2000s. So we don't have to rely on such synthetic, simple landscapes anymore. We can actually work with landscapes that are constructed from experimental data. So that's what we set out to do. And this is how we move into part 2 here. So this is a paper that we published in PLOS Comp Bio a couple of years ago now. And Alejandro was aware of some work that I did during my postdoc where I constructed more than 1,000 empirical adaptive landscapes. So these landscapes pertain to gene regulation. And the way that they're constructed, we have each landscape corresponds to a transcription factor. So a transcription factor is a protein that binds DNA to regulate gene expression. And these landscapes are constructed such that the genotypes in the landscapes are DNA sequences. These are sequences that the transcription factor binds. And then the elevation of each of these genotypes, or what defines the topography of the landscape, is the strength with which that DNA sequence binds a transcription factor. So in constructing these landscapes, we could study selection for increased binding affinity of transcription factors to DNA. All of those details really aren't that important for today's talk. All that you need to know is that we have more than 1,000 empirically characterized adaptive landscapes. And what Alejandro wondered was whether or not these landscapes themselves exhibited some sort of bias in the mutations that they contained. So to remind you about this classification of mutations as either transitions or transversions, transitions are between a purine and a purine, or between a pyrimidine and a pyrimidine. Transversions are between a purine and a pyrimidine, or vice versa, right? And the naive expectation is that you would observe one transition for every 2 transversions. Now, what Alejandro wondered was whether or not these landscapes themselves had some sort of a bias towards transitions or to transversions. To figure that out, or let me just illustrate that quickly. Let's say you have some adaptive trajectory in this landscape that starts here. The first step could be a transition, and the subsequent steps might be transversions. In which case, you could say that that adaptive trajectory had an enrichment for transversion mutations over transition mutations. Right? But to quantify this, what Alejandro did is he created a measure that he called composition bias. So this measure is on the unit interval. When it equals 0.5, that means that you observe 1 transition for every 2 transversions. If it's greater than 0.5, that means that you observe more transitions than that null expectation would say. And if it's less than 0.5, you observe more transversions than the null expectation. So the first thing that he did is he just calculated the composition bias of these entire landscapes. So for each landscape, he looked at all mutations in the landscape, and he asked, what's the composition bias of that landscape? And he did that for the more than 1,000 landscapes that we had constructed. And what he found was actually totally uninteresting. It was a negative result. This was like, okay, these landscapes are just exactly behaving according to our null expectation. 1 transition for every 2 transversions. However, if you instead look at what we call mutationally accessible paths, so this is like what I showed you a moment ago, it's just an uphill path, the landscape, so just a path in which adaptation keeps happening as you move up that path, you see a totally different story. So here, you see an enrichment for transversion mutations. So that means if you just look at the paths from some random location in the landscape to the global adaptive peak, you're more likely than expected to find transversion mutations along that path. And this is interesting because, as I showed you before, mutation bias is in the opposite direction, right? It's towards transitions. So what Alejandro wondered is whether or not there might be some interaction between mutation bias toward transitions and this composition bias towards transversions. Like, might that frustrate adaptive evolution, or might it facilitate adaptive evolution? So to figure out the answer to that question, first I just want to highlight that these transcription factors belong to loads of different protein families, and what we were observing was not just like 1 or 2 protein families that were driving our trends. Rather, the majority of the protein families that we looked at had this bias in their accessible mutational paths towards transversions. Okay. Right. So now he wants to know, is there some interaction between mutation bias and composition bias that influences adaptive evolution? So what you're looking at here, first of all, you're looking at data for 16 of the landscapes that we studied. So you'll see all of them in a moment. These are just 16. And these 16 are chosen because they have a strong composition bias towards transversion. So that's what that upper rectangle is. So you see that dot is way to the left. That means that these landscapes all exhibit a very strong composition bias towards transversions. Now, in the main panel on the y-axis, you're looking at the probability with which the evolving population reached the global adaptive peak in relation to what's shown on the x-axis, which is mutation bias. So this is just a knob you can tune with respect to the generation of genetic variation. So are you introducing more transitions or more transversions, right? So this is defined on the unit interval, just like composition bias was. And what this figure reveals is that the probability with which the evolving population reaches the global adaptive peak is maximized when the mutation bias is aligned with the composition bias, right? So when these two things are in sync, the population is way better able to get to the global adaptive peak. And if we then look at this same plot, but for all of the different landscapes, again, grouped according to their composition bias, so we just broke them up into these 5 bins, and like where in the composition bias spectrum they reside is shown in the upper rectangle, you see that this is a consistent trend. So whenever mutation bias is aligned with composition bias, the probability that the evolving population reaches the global adaptive peak is increased. And when these things are anti-aligned, that probability decreases. So this is just one of many results from this paper that suggest that mutation bias can interact with composition bias to influence adaptive evolution. We also found that this interaction influences the predictability of the evolutionary process and the evolution of mutational robustness. Now, for those of you in the audience that aren't modelers, maybe you work with experimental systems, you're probably looking at this plot and thinking like, oh, that's cute, like, look, but like those lines are just so clearly from simulations, right? Like that real biological data never looks that clean. So, what kind of evidence do we have that mutation bias could influence adaptive evolution in nature? That brings us to the next paper. So this was a collaboration with Professor Alex Hall's group at ETH and Professor Sebastian Gagnew's group at the Swiss Tropical and Public Health Institute in Basel. We decided that we wanted to study whether or not transition bias could influence adaptive evolution in the bacterium Mycobacterium tuberculosis. So Mycobacterium tuberculosis is the causative agent of tuberculosis, which is, an absolute killer. And we chose to work with this species for several reasons. First of all, we know that it has a strong mutation bias towards transitions. So I showed you this plot before, but what I didn't point out is that one of these 5 bacteria is Mycobacterium tuberculosis. So it has this really strong bias towards transitions in the generation of new genetic variation. Moreover, the population genetic conditions of Mycobacterium tuberculosis are conducive to seeing mutation bias influence adaptive evolution, because the populations have very small, they go through massive bottlenecks upon transmission. So Mycobacterium tuberculosis it goes through spit, right? So they like these little water droplets. So they're these tiny populations that go from one human to the other. And Mycobacterium tuberculosis has one of the lowest mutation rates of all bacteria. So you put these things together, and that means that the mutation supply is very low, right? And so that means that that Mycobacterium tuberculosis is likely to undergo these sort of origin fixation dynamics that I talked about before. So evolution most likely doesn't proceed from abundant standing variation, but rather from new mutation. Now, the other reason that we chose to work with Mycobacterium tuberculosis is that, unlike many other bacteria, it evolves antibiotic resistance or any kind of adaptation exclusively via chromosomal mutation. So it doesn't rely on horizontal gene transfer, it doesn't rely on plasmids, nothing like that. It is just de novo mutation in the chromosome. Additionally, this is one of the few species where we thought we could get bona fide, a data set of bona fide adaptive mutations. And the reason we thought that is, since it's such a worldwide health problem, there's just been a massive effort at understanding which mutations confer antibiotic resistance, right? And this is important because if you want to do a study where you're trying to say that a particular mutation bias like creates an enrichment for a mutation type in a set of adaptive mutations, first of all, you need to know darn well that those mutations are adaptive. And second of all, you need to have lots and lots of those mutations in order to statistically power your tests. So for all of these reasons, Mycobacterium tuberculosis was kind of like the perfect setup for studying whether or not mutation bias can influence adaptive evolution. So, as I said, this is a collaboration with Sebastian Gagnew's team, and in particular with one of his postdocs at the time, Fabrizio Minardo, who was kind enough to curate a list of bona fide adaptive mutations in Mycobacterium tuberculosis. These are mutations that are known experimentally to confer resistance to antibiotics, and that appeared in at least one of these roughly 9,000 Mycobacterium tuberculosis genomes that we had publicly available. Importantly, these are clinical isolates, right? So this is evolution in the wild. This is actually happening in the human lung. Now, this was at that time, and still today actually, the largest data set of bona fide adaptive mutations. In total, we had 2,775 mutational events across 152 mutational paths. And I'll explain this distinction between paths and events right now. So a mutational path, for example, is this C to G transversion in the 315th codon of the catalase CatG that changes serine to threonine. That is a mutational path. Now, this particular mutational path confers resistance to isoniazid. And when we did a phylogenetic reconstruction of these roughly 9,000 MTP genomes, we were able to identify how often this mutational path was taken independently in that phylogeny. And we found that there were 445 such mutational events in that phylogeny. So for this particular mutational path, we observed 445 events, right? So these are two sort of ways that we can work with our data at the level of paths and at the level of events. Okay, let's see what we found. Now, you're looking at actually two data sets here. The data set that we curated is called the Basel Data Set. And then while we were working on this, another group published a list of bona fide resistance-conferring mutations in MTB that was really wonderful for us because we could then use them as kind of like an independent data set to look for what we're looking for in our Basel data set. So do we see the same thing basically in these two independently curated data sets? And indeed we do. So the x-axis has our data grouped according to paths and events. And on the y-axis, we have this ratio of transitions to transversions. And again, remember, our null expectation is that we would observe one transition for every two transversions. And for both data sets and for both classifications of our data according to paths and events, we find a significant enrichment of transitions. Right, so this is pretty strong evidence that this bias in the generation of genetic variation actually influences which genetic changes drive adaptation. However, you could think, well, maybe there's some like selective advantage of transitions relative to transversions. That's a possibility. So for example, at the amino acid level, given the standard genetic code, we know that transition mutations are sort of more gentle in that they cause smaller changes to the physicochemical properties of amino acids than do transversion mutations. So maybe this is all selection. It has nothing to do with mutation, right? So how do we rule out that possibility? Well, one way is to look at a subset of the data where these mutations actually didn't cause changes in proteins. These are non-coding mutations, mutations that are either in promoters or in the 16S ribosomal gene RRS, which is not translated to protein. And here we see an even stronger enrichment of transitions. And this is actually one of the few like real question marks of this study. I do not know why it's so much stronger here in the non-coding region than it is in the coding regions. But nonetheless, this allows us to certainly rule out any kind of selective advantage of transitions, at least at the amino acid level. Okay, so this was really clear-cut evidence that mutation bias, in particular transition bias, can influence adaptive evolution in nature. Now, moving into the final part, which will appear, I believe, on Tuesday in PNAS. So the study that I just described and other related studies, they're sort of based on statistical tests of asymmetry, right? So they're like saying, let's group the mutations according to something like transitions versus transversions, and then let's compare that to some null expectation. Say our null expectation is one transition for every two transversions, and then just come up with some statistical test of asymmetry. Like, do we see that there's some enrichment relative to this null expectation? But what we were wondering is about the more general case, like what if we just look at the entire mutation spectrum and ask, to what extent does the entire mutation spectrum, which simultaneously includes all kinds of mutation biases, to what extent does that spectrum shape what we call the spectrum of adaptive substitution? So this is just the spectrum of mutational changes that underlie adaptation. So let me explain this in a bit more detail now. So to answer this question, we created a new modeling framework, and this is a nice modeling framework because it can be used in other systems than the systems that we used it here in this study, and we hope this will be a resource for the scientific community. So as I said, our goal is to model the spectrum of adaptive substitutions, which can be defined in a variety of ways, but the way that we define it in this study is a vector of substitutions from a given codon C to an amino acid A. So given the standard genetic code, there are 354 such changes. And this spectrum just tallies the number of those changes that we see in some experimental data set, like the data set that I just described in the previous study. Now, we want to model this is a distribution, right? We want to model this distribution as being directly proportional to the frequency with which the codon appears in the genome, right? Like you would just expect more mutations from a given codon if that codon is really frequent in a genome. So that's clear. So we want to model this distribution as being directly proportional to the frequency of the codon, and then potentially proportional to the mutation rate from codon C to amino acid A. And by potentially proportional, what I mean is that we're going to exponentiate this term with beta, And beta is our free parameter in this model. So this is what we're going to use to fit our data. So the spectrum of adaptive substitutions, this is something that we populate with experimental data. I'll show this in a moment. The frequency with which you observe a codon, like you could just populate that vector from genome sequences. And then these mutation rates you can populate from the sort of data that I showed at the beginning of the talk, these like fluctuation assays, for instance, that tell you the rate with which given mutations happen in the absence of selection. So all three of these elements of this model are constructed from empirical data, right? And then you just have this one free parameter beta that you use to fit the model. And if beta is equal to 0, what happens? This term just drops out, right? Mutation rates don't matter at all. But if beta is 1, then this means that there's a proportional influence of the mutation rate on the spectrum of adaptive substitution. And values of beta between 0 and 1 indicate an intermediate influence of the mutation rates on the spectrum of adaptive substitutions. Okay, so this is our modeling framework. It is a phenomenological model. It is not based on PopGen or anything like that. It is just, sorry, my daughter just got home. Yes, you can have that. It is not based on population genetics or anything like that. It's just saying, hey, to what extent can we model this distribution of genetic changes that underlie adaptation, just knowing these two other things, like just knowing how often these codons occur in the genome and knowing mutation rates from codons to amino acids. That's all. So here's the workflow. You've got to have data, right? So you're starting with data. The data that we're working with are either from laboratory evolution experiments or from clinical isolates. So the clinical isolates are the Mycobacterium tuberculosis data I told you about before. From this, you can generate a list of adaptive substitutions. From that, you can populate this spectrum of adaptive substitutions. And then you can fit this model. So this is the same model I showed before. It's just that we took the log of both sides to get rid of this proportionality business. And then once you have that fitted model, you can try to make predictions. You can try to predict the observed spectrum of adaptive substitutions. And importantly, I just want to reiterate that all three components of this model come from empirical data, right? We're just fitting data. That's all we're doing here. Okay, so let's talk about these data a bit. So we have 3 study species. These are all microbes. Saccharomyces cerevisiae, we have data from 4 studies using laboratory evolution experiments under different stresses. So high salinity, low glucose, rich media, gene knockout. E. coli, these data come from a single study of laboratory evolution under temperature stress. And the Mycobacterium tuberculosis data I've already described. So these are our 3 study species. Now let's look at some of the components of this model. So the first component is the mutation spectrum. So here again, we've seen this throughout this talk. On the y-axis, we have the relative rates of these mutation types, grouped according to the 6 types of mutations and further grouped according to whether they're transitions or transversions. And you can see there's heterogeneity here, right? So both within a species, some mutation types occur more often than others. And between species, some have a higher rate of G to A than do others, right? And you can see that more clearly if you actually do all these pairwise comparisons here. So for instance, the height of this bar here shows that there's a 2.5-fold increase of A to T transversions in Saccharomyces cerevisiae relative to Mycobacterium tuberculosis. So, when you're looking at that big plot on the left, you might think, oh, those bar heights, they're not that big of a deal. They're not that much. But if you actually do the fold change, like you compute that, you're talking like sometimes 2 or 3 X differences. Okay, so there's really differences in these 3 mutation spectra. So are these differences borne out in the kinds of genetic changes that drive adaptation? Our first observation, this is kind of a very simple, just having fun kind of first glance at our data. If we just take all of the mutations that are in our data set and we classify them according to their 6 mutation types, and we just calculate their frequency, that's what's on the x-axis. And on the y-axis, we have these mutation spectra in this main panel, right? So that's what's on the x-axis. So we just correlate, like how often do we see a given mutation type against how often do they happen in this mutation spectra. We see this really tight correlation in all 3 species. So this is already some pretty nice evidence that mutation bias is influencing which genetic changes drive adaptation. But this is a very naive analysis because it doesn't take into consideration codon frequencies. It doesn't take into consideration the structure of the genetic code, just nothing like that, right? But still pretty cool to look at. And these codon frequencies, these are really important to take into consideration. So here, if you look at the frequencies of all the 64 codons in the different species, you'll see that these different species use different codons quite differently, right? So for instance, GAA is the most frequent codon in yeast, but the second most in E. coli and appears far less frequently in Mycobacterium tuberculosis. So like in yeast and E. coli, we might just expect to see more mutations from GAA just because there's more codons from GAA. So we want to use this model that I told you about to actually take this stuff into consideration. So here again is that model. And what we're trying to fit here is this beta term. This measures the extent to which the mutation rate is influencing the spectrum of adaptive substitutions. And remember, its maximal influence, this proportional influence, occurs when beta equals 1. And what we find is that in all three species, the 95% confidence interval on beta contains 1 and it is statistically significant, right? So and importantly, beta, these 95% confidence intervals never contain 0. So this is clear-cut evidence that mutation bias has a proportional influence on which genetic changes drive adaptation in these three species. Now, I was really struck by this result, and my first reaction was like, well, what happens if we just throw in some random mutation spectrum? Like, how often do we just do this good by chance alone? So we asked that question by generating thousands of random mutation spectra for these three species, and then just refitting our models, and then comparing the log likelihood of these different model fits. So in each one of these panels, this dashed line and what's right of the dashed line, that indicates how often a randomly generated mutation spectra does better than the empirical mutation spectrum. And we find that almost never happens, right? Less than 4% of the time do random mutation spectrum provide better model fits than the empirical mutation spectrum. And you might think like, well, that's kind of obvious, but like consider this, right? These mutation spectra are generated in controlled laboratory conditions, right? These are based on like fluctuation assays. Yet we are finding that they can tell us about which genetic changes drive adaptation in the lab and in nature. So in totally different conditions, right? So this really highlights the relevance and importance of characterizing these mutation spectra. Finally, if we use the spectrum of adaptive substitutions to infer what the optimal mutation spectra would be, right? So now it's like the mutation term is actually what we're learning in our model. And we then plot that optimal mutation spectrum against the real empirically characterized mutation spectrum. Again, we have these super tight fits, right? Which is saying to us that these empirical mutation spectrum are just spot on. You really can't do better than the empirical spectrum. So that's also a pretty cool result. Okay, then finally, I talked about prediction. I'm almost done here. So we find that the mutation term has its maximally theoretically predicted influence on the spectrum of adaptive substitutions, right? It always contains 1. But what does that tell us about our ability to actually predict the spectrum of adaptive substitutions? Now we have a fitted model. Can we make predictions about which genetic changes drive adaptation? And we find that our ability to do that actually varies significantly amongst these three species. So in Saccharomyces cerevisiae, we're able to do pretty good. You know, there's a Pearson's correlation of 0.68. E. coli is like, not so great. And then Mycobacterium tuberculosis is just abysmal, right? We can't make predictions there, even though our model is telling us that mutation bias really matters, but we just can't use it to make predictions. And a big part of this paper is actually then following up on these analyses that are based on empirical data with computational simulations to try to understand like the population genetic conditions, the demographic conditions that cause this variability in prediction. And if you're interested in that, I recommend you to check out the paper that's coming out next week. I'm not going to go into those modeling details now. Okay, so I think I was just within 45 minutes. that's what I've got today. I hope you take something away from this and enjoyed it. And I'm here now to have a chat.
Yeah. Firstly, thank you very much for your presentation, Professor Payne. Not surprisingly, it was very informative and explanatory. So now our team has compiled the questions from the chat. So if you are okay with it too, they can forward them to you without taking so much of your time.
Great.
Okay, then I think who comes first?
Okay, I can ask my question. This is actually like a personal question for me. So maybe most of our viewers don't know about it, but you actually studied computer science and mathematics in university. And as a computer science major myself, I wonder why you have leaned towards evolution and biology for your PhD. What was the turning point in that, or what caught your interest in the first place?
Yeah, happy to answer that question. So my PhD was mainly focused on this, which was in computer science, as you point out, was mainly focused on this class of optimization algorithms that are inspired by Darwinian evolution. They're called genetic algorithms, which, as a computer scientist, I'm sure you've come across. And I just thought that was so cool, that you could do evolution in the computer. And I was obsessed. I spent an inordinate amount of time writing these evolutionary simulations and studying their dynamics. And I guess over time, I kind of came to the realization that it wasn't solving optimization problems that inspired me. I didn't care that I could solve the knapsack problem faster by a few decimal points than the last guy. I don't care. But what did fascinate me is just the evolutionary process itself. It's so simple. It's such a simple algorithm, but it's responsible for all of the richness of life that we have around us. And that just twisted my mind. How is this trivial algorithm causative of everything around us? And that question just led me into evolutionary biology, and I started to find that the stuff of life, like DNA molecules, RNA molecules, proteins, are just so much more interesting than zeros and ones. And so, don't get me wrong, I'm not bagging computer science at all. I love computer science. I still, almost all of my work is computational in nature. I'm so happy that I trained in computer science. But I'm also so happy that I made the switch.
Okay, thank you. It was an inspiring talk.
I'm glad you enjoyed it.
I think Nevzat can ask the second question if he's also available.
Great.
Why did you prefer the negative logarithmic regression over the linear regression in your model?
Why did we use negative binomial regression?
Yes, over the linear regression model.
Yeah, good. I didn't talk about that. Thank you. So the reason is that the counts data that we're trying to model are over dispersed. So the first thing that we thought of is we could use Poisson regression actually, but that just falls flat with the kind of distribution that our data take. So it's a distribution dependent decision that we made there.
Okay, thanks.
Thank you.
Since the real reason and mechanism of codon optimization and codon bias cannot be fully revealed, isn't this factor interrupt and disturb your studies a lot? How do you deal with that problem, Professor Payne?
Sorry, could you please repeat that a bit slower?
Okay, sorry.
It's okay.
Since the real reason and mechanism of codon optimization and codon bias cannot be fully revealed, isn't this factor interrupt and disturb your studies a lot? How do you deal with that problem?
So you're talking about codon optimization, with regards to translational efficiency, say?
Yes.
Yeah, so I wouldn't say that it does interfere with our problem. So at least in the modeling that we do, the codon frequencies are a part of that model, right? So if those codon frequencies are the result of some sort of optimization process for translational efficiency, then that's already baked in. But then if you're talking more generally about the choice of synonymous codons, I mean, I don't see it as problematic. So I see where you're going with this, and I think it's a really interesting thing to think about. I love thinking about codon bias because it's synonymous but not the same. It's such a cool thing to think about. But so let's say we're just talking about one codon, right? And there's some selective pressure for translational efficiency such that one of the synonymous codons is better than the other. I mean, it's still mutation that is going to bring you there, right? So I think that whatever selective advantage you have of one synonymous codon over another, it's no different from the selective advantage of one amino acid over another. Does that help?
Okay, thank you.
You're welcome. Okay, so now you really got me thinking here. This is cool. So in our modeling framework, however, so we're always doing codon to amino acid changes. So we're actually never modeling mutations amongst synonymous codons by definition, because they don't change amino acids. And that could be a really cool thing to do. I think the challenge there would be getting a good data sets of bona fide adaptive mutations amongst synonymous codons, because that's harder to identify, But that could be really cool if you could find a data set like that. I think it'd be fun to apply this model to that data set. Good.
Yeah, also, it was good. Actually, it was my question when I was listening to your presentation, because I never think like a computational or empirical scientist when I am doing my own research. I'm more of a experimentalist rather than a theorist. So
Okay. Yeah. Good.
I was actually ignoring the fact that you are doing what you do on models and empirical data. So yeah, but I think it's also an interesting point in that too. It's kind of a cross section between them. So I was interested in that. Yeah. Okay.
Yeah, I think it's a great place to operate. Theory is wonderful. Data is wonderful, or data are wonderful, I should say. And but what's the best is when you can get those two to talk to each other.
Mm-hmm.
So I love working at that interface that you're describing. And and if it weren't for people like you doing real experiments, generating real data, I'd have nothing to work with. So thank you.
Yeah. Okay. We thank you for your work on interpreting the data that we come up with and make it meaningful, I think, because there's a lot of data and few people to interpret it. So yeah. Okay.
Yeah, this is what I always tell my students, with how much data is generated these days. Mm-hmm. It's just necessary that those data have answers to questions that no one has thought to ask. Mm-hmm. There's just so much data out there that I think now it's just about being clever. It's just about thinking okay, how do I combine data sets or look at a data set from a different angle to address a question that the people who generated the data never thought to ask.
Yeah, it's knowledge is power, but you have to somehow categorize and classify and interpret that knowledge to actually have something in these days. So yeah, it's important to do it in a in a safe manner and also efficient manner.
Yeah, agreed.
Yeah. Maybe Dahan can ask the last question to you.
Yeah, okay, I will ask it. Why does Mycobacterium tuberculosis only adapt chromosal mutation? And are there any specific reason why this species cannot adapt HGT? Is this also related to its evolution path?
Sorry, the last part I lost you. Why does it not have what?
Why this species cannot adapt HGT? And is this also
horizontal gene transfer?
Yeah.
Yeah. So why doesn't it use plasmids? Why doesn't it use horizontal gene transfer? I have to just reveal my ignorance here. I do not know the answer to that question. And this, I think, speaks sort of to the power of collaboration. So all of the domain expertise regarding Mycobacterium tuberculosis on that project came from Sebastian Gagnew's group. So they are really some of the leading experts in the world on Mycobacterium tuberculosis and antibiotic resistance in that pathogen. So I regret that I cannot give you an answer, but thank you for the question because I want to know the answer and I will look it up after this talk.
Thank you.
I think because of the limited time, we need to finish our talk in here. So thank you again, Professor Payne. We deeply appreciate your talk and your contribution to the series. Hope to see you later in another talk.
I hope so as well, and thank you all for the invitation. It was a pleasure, and I wish you all a very nice afternoon.
See you.
Bye-bye. Thanks.
Bye.